Good morning. It's September 11th, and this is your daily brief in science. Here's everything you need to know. A new study published in Nature establishes a direct link between major fossil fuel companies and the increased likelihood and severity of heat waves. The research shows that nearly a quarter of heat waves from the year 2000 to 2023 can be directly attributed to emissions from specific corporations. Human activities have made all examined heat waves significantly more probable, with some becoming 10,000 times more likely due to climate change. Without human influence, such events would be virtually impossible. The study emphasizes that individual contributions to climate change are substantial enough to impact specific extreme weather events, countering common arguments to the contrary. Led by an international team including scientists from the University of Leipzig and Columbia University, the research uses climate modeling and historical emission data. The analysis considers major emitters such as China, India, ExxonMobil, and Shell. It involves comparing climate models with and without greenhouse gases to assess human impact on heatwave frequency and severity. The findings support the development of frameworks for attributing extreme weather to human activity, which could influence climate policy and legal accountability. Despite ongoing legal challenges, this study provides critical evidence to hold fossil fuel companies accountable for their role in climate change. It builds on principles from the 2023 International Court of Justice Advisory Opinion, which states that countries failing to address climate change may be liable for reparations. Limitations like underreporting in regions such as Africa and South America suggest the true impact may be even greater. The research underscores the need for global emission reductions and countering climate disinformation. It also aims to simplify legal actions against large emitters, reinforcing liability for damages and climate adaptation costs. A new push to develop cancer vaccines could transform treatment options for breast cancer patients. The Cancer Vaccine Coalition, founded by breast cancer survivor Kristen Dahlgren, is working to accelerate vaccine development and approval in the U.S. while highlighting international progress in large-scale trials. Cancer vaccines are nearing a critical point, shifting toward less toxic and more effective therapies, according to Dahlgren. One of the most advanced vaccines, WOKVAC, has shown a favorable safety profile and strong immune responses in early-stage HER2-positive breast cancer patients. The coalition's funding expansion aims to enroll more participants to better understand the vaccine's immune-stimulating capabilities and improve the robustness of the trial. A $500,000 grant from the coalition and the V Foundation will support the University of Washington's clinical trial expansion. The trial involves administering WOKVAC alongside chemotherapy and targeted therapies before surgery, with the goal of boosting immune response and creating long-lasting immunity. Led by Dr. Nora Desis and Dr. Will Gwynn, the study focuses on harnessing the immune system to prevent recurrence and improve long-term outcomes. The trial's expansion is further supported by a $2 million matching gift from the Brian and Sheila Jellison Family Foundation, emphasizing its potential impact. Patient testimonies such as Carmel Lorino's reflect hope and perceived protection, highlighting the vaccine's promise to save lives. WOKVAC targets HER2 and other cancer proteins to stimulate tumor-specific immunity, addressing the needs of over 280,000 women diagnosed annually with breast cancer, especially the aggressive HER2-positive subtype. Scientists at Columbia University's Zuckerman Institute have identified a previously unknown brain region in mice called the bed nucleus of the stria terminalis, or BNST. This region acts as a master regulator of feeding behavior, influencing the consumption of sugars, fats, salts, and other dietary components. The discovery was made using advanced techniques such as optogenetics and anatomical tracing, which mapped the BNST's role and connections within the brain. When activated, BNST neurons cause mice to eat even when they are full and to consume inedible objects like plastic pellets, demonstrating its powerful control over feeding. The BNST integrates signals related to hunger, nutritional deficits, and food pleasure, serving as a key hub linking sensory food traits to weight regulation. Experiments show that more BNST neurons respond to sweet and salty tastes in hungry or salt-depleted mice compared to satiated ones, indicating a role in hunger and nutrient signal processing. The study, published in Cell, also identified neurons in the central amygdala connected to the BNST, which influence feeding by either amplifying or suppressing the desire to eat. Activation of BNST neurons increases food consumption, especially when taste signals like sweetness stimulate neurons in the amygdala that then activate the BNST. The findings suggest that targeting the BNST could have therapeutic potential for conditions like cachexia, where patients experience severe appetite loss and weight decline during chemotherapy. Additionally, the research revealed anatomical connections between the BNST and other brain circuits that monitor internal states such as hunger and salt deficiency. Notably, the weight loss drug semaglutide targets neurons in the BNST, providing insights into its mechanism and opening possibilities for developing appetite-suppressing therapies with fewer side effects. Researchers at the University of Pennsylvania have created a new photonic system that guides light through crystalline structures using principles from topological physics. This system forms a secret tunnel for photons, making their travel resistant to imperfections, such as bumps and bends. The key innovation involves using circularly polarized lasers to induce a Floquet topological phase in the material. Experiments confirmed that this polarization opens a full energy gap with a topological Chern number of 1, resulting in protected one-way edge states. These states enable light to travel unidirectionally along the edges of the material, even when defects are present. Spectroscopic measurements validated the creation of a topologically non-trivial band gap, supporting robust photon flow. This development moves the concept from theoretical models toward practical, scalable photonic devices. The researchers plan to extend this approach to three-dimensional crystals and microwave frequencies with potential applications in quantum information protection and secure communications. Future work also aims to explore the protection of fragile quantum states of light, which could impact quantum computing and sensing. The system employs nonlinear optical interactions, such as frequency doubling, for advanced information processing. This technology could lead to stable optical isolators, lasers, and integrated photonic devices that do not rely on magnetic components. Despite challenges from the COVID-19 pandemic, the team stabilized their system by 2022 through collaboration and precise control of ultrafast lasers. The research was published in Nature Nanotechnology, highlighting the creation of chiral edge states that allow photons to move in a one-way path, even with imperfections. Support from U.S. agencies underscores the strategic importance of advancing photonics and quantum technologies, inspired by studies on electronic topological insulators. Intensity Therapeutics has announced early positive results from its Invincible-4 trial, which tests the intratumoral therapy INT230-6 in treating pre-surgical triple-negative breast cancer. The trial involves administering 2 doses of INT230-6, spaced 8 days apart, followed by standard immunochemotherapy. Initial signs include tumor necrosis and immune activation, suggesting potential effectiveness. The primary goal is to improve the pathological complete response rate, a key indicator of long-term outcomes. The study is a multi-center, open-label trial authorized by Swissmedic and the European Medicines Agency, conducted with French and Swiss cancer research groups. It plans to enroll up to 54 patients, comparing INT230-6 plus standard therapy versus standard therapy alone. INT230-6 is a proprietary formulation combining cisplatin, vinblastine sulfate, and a diffusion enhancer, designed to kill tumors and stimulate immune responses without causing immunosuppression. While early safety data in Cohort A is favorable, some patients experienced localized skin irritation, leading to a temporary pause in new enrollments. The pause was voluntary and not mandated by regulators, with plans to resume with a modified dosing regimen. The company remains optimistic about improving safety and efficacy, aiming to turn deadly cancers into manageable conditions. Beyond this trial, INT230-6 has been studied in metastatic and locally advanced cancers, with additional trials planned, including a Phase 3 in soft tissue sarcoma. Despite safety concerns, experts see potential in INT230-6 to improve treatment outcomes and cosmetic results, focusing on increasing complete tumor responses. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.